Hello. On this beautiful day, we're outside to talk about plants, specifically vascular plants. Most of the plants you see here, they're all vascular plants.
Wait, so is that moss a vascular plant?
Moss actually isn't, because it does not have a vascular system, which means they do not have xylem and phloem associated with it.
Wait, what is xylem and phloem?
Do not worry about that yet, we will teach you. It should be noted though that without vascular tissues, these plants lack true stems and roots.
As we mentioned, this tree has vascular tissues, the xylem and phloem. The xylem is responsible for water uptake, whereas the phloem is responsible for sugar and nutrient uptake.
Now let's move on inside to get a better understanding of things. Since you only know the basics, let me teach you from the beginning. During the globular embryo stage of a plant, there are a few initial cells. The early globular embryo consists of 3 cell layers, the first one being precursors of epidermal, the second one being ground, and the third one being vascular cells. However, we will only focus on vascular in this project. In fact, it is believed that all vascular tissues are derived from 4 initial cells.
I assume the provascular cell layer is located in the center of the embryo. So do these cells divide and differentiate during embryogenesis to give rise to provascular tissue?
Exactly. And these precursor cells eventually make xylem and phloem. Something interesting is that vascular cells, marker cells, which is called SHR, are expressed before globular embryo formation. Therefore, provascular cell identity is determined in early embryogenesis.
Are SHRs all that is needed for us to make xylem and phloem?
No, as you can see in this image, auxin is needed for stem cell initiation or specialization and help with cell proliferation, which eventually leads to cell differentiation. Clearly, other hormones such as cytokinin and gibberellin also plays a role in this process, but auxin is required for stem cell initiation.
So what happens when auxin is absent?
Loss of function of auxin response transcription factor, which is called MP or ARF5, leads to defect in asymmetric cell division in the part of the globular embryo, which eventually leads to a loss of vascular cells. Furthermore, in auxin response pathway, transcriptional target genes, specifically TMO5, encodes transcription factors that are expressed in all 4 initial vascular cells, but expression is eventually restricted in 2 cells facing each other, which are the xylem initial cells that eventually form the xylem axis.
Now we know a basic mechanistic understanding of vascular development, so let's try to get a better understanding of the development of xylem. As we mentioned above, the xylem is responsible for water transport. Obviously, these tall trees need the xylem to be able to transport water all the way from the soil to the top of the plant. The xylem is made up of 4 elements, including tracheids, vessel elements, parenchyma, and fibers. The xylem is made up of dead cells. The first type of xylem tissue that evolved are tracheids, which are responsible for transporting water. The cells are elongated, tapered, and connected through pits. Vessel elements evolved after tracheids, and they are larger and more efficient in their water transportation. There are 2 types of primary xylem, protoxylem and metaxylem. Protoxylem is the first forming xylem tissue, and it consists of elongated cells with thin walls. This makes it very stretchable, allowing less rigid growth of the plant. Then, the later maturing primary xylem is called the metaxylem. It consists of wider, shorter cells with thicker walls, meaning it's more rigid than protoxylem. As the cell stops elongating, the cavities of vessel elements and tracheids get larger and become more specialized. Thus, the wider lumen of the metaxylem cell allows for more efficient and rapid transport of water and minerals.
So is there any mechanisms that is directing the formation of metaxylem versus protoxylem?
Yeah, so transcription factors named vascular-related NAC domain 6 and 7 play an essential role in xylem vessel differentiation. It has been reported that in Arabidopsis, The VND6 promotes differentiation in the metaxylem, whereas VND7 induces differentiation in the protoxylem.
Okay, so now I understand what xylem is, but how does it work?
The way in which the xylem is able to transport water against the force of gravity is through evaporation, which in plants is called transpiration. It incorporates a suction force that allows for transport as well as provides like a cooling source for the plant. What happens is the water reaches the cells at the end of the leaves, and at the end of the leaves there are these pores called stomata, which when they're open, it allows for water to evaporate into the atmosphere. This creates a suction force from the bottom of the tree to the top. This is also why xylem walls need to be thick in order to withstand that suction force, which in turn helps explain why xylem helps with the structural support of the plant. But we'll get into that later.
The other main tissue is the phloem, which is the photosynthate conducting tissue. It is composed of CF tubes, elements, and companion cells.
Do sugars not move along the same tube as water? I would have thought the xylem was enough for everything.
No, actually, if you remember, xylem moves from bottom up, while phloem moves in both directions. Unlike xylem development, our knowledge about the molecular control of phloem formation is still relatively poor, although no differentiation-inducing factor has yet been found to trigger phloem-like differentiation. It has been found that auxin and other hormones may play an important role in phloem cell differentiation.
Okay, I see. So does it work the same way as the xylem?
It actually worked through a process called translocation, which simply describes the transport of food and organic materials up and down within the plant. So we have 4 basic steps that describe this process. It starts with sugar that got loaded into the phloem tube, which is an active process and requires energy. Hence, phloem cells are living. Then the concentration of phloem tubes increases, and this is where unloading comes in. Through osmosis, water from xylem flows into the phloem to decrease the increased concentration of sugars. However, there is a problem when the water flows from xylem to phloem, since it causes the phloem tube to swell up. There is a lot of pressure coming in, resulting in the sap to move from higher to lower concentration of sugar, which is the lower part of the cell that also contains less pressure, thus releasing that pressure. This is referred to as bulk flow.
Wow, I'm so glad that now I know how xylem and a phloem work.
It's important to note that there is secondary vascular tissue growth, which is usually a process in woody plants. The vascular cambium is a meristematic tissue that's responsible for deciding the amount of secondary phloem and xylem that gets produced. These new cells are then able to form into concentric rings, which in turn results in a greater girth of the stem or trunk. Auxin concentration plays a key role in the regulation of cambial cell divisions and elongation thickness of the cambial layer. The secondary growth allows for the stem to grow in girth and aids in the water transport. Let's say there's a lack of xylem. How would these 100-foot trees grow to be so tall? They wouldn't. The plants are constantly fighting against gravity, and without xylem maintaining tugger pressure, plants would not be able to grow tall.
The vascular tissue allows for more structural support, so plants like trees are able to grow tall and strong. The xylem tissue provides more mechanical support and stands upright even in tough environmental conditions.
An elimination of the vascular tissues would have devastating biological consequences. The evolution of vascular plants also increased oxygenation in the atmosphere, which had a significant impact on the evolution of animals. Animals were able to grow larger bodies, more complex respiratory systems as a result of the elevated oxygen.
I hope you enjoyed our little lesson on vascular tissue development and understand the significance of them.
Our world wouldn't be the same without them.